Bismillahirrahmanirrahim. Welcome to my YouTube channel, Science for Everyone. Today I will discuss a very important topic with you people, and the name of the topic is mitochondrial DNA. In this part of the video, I will give an introduction of mitochondrial DNA, and I will discuss that what is the difference between the normal cell DNA and the mitochondrial DNA. In the second part of the video, I will discuss the replication of mitochondrial DNA. Okay, so why I'm discussing this mitochondrial DNA, because I want to tell you that in our cell there is an organelle that is known as mitochondria, and it is very much important because it is known as the powerhouse of the cell, and the respiration is taking place inside the mitochondria. These mitochondria are very much different from the other organelles because it contains its own DNA. So because of this feature, it is very much unique in nature, and it can also replicate by itself. So that's why I have chosen this topic. So let's start with the video lecture. First of all, I want to tell you there is a question in mind that how does it originate? This mitochondria originates because it contains its own DNA. So how does it enters the cell? And so these such type of questions come in the mind. So I want to tell you that long ago, about millions and billion years ago, as we know that there was no oxygen on the earth. And after that, the production of oxygen by the cyanobacteria, within million years, the oxygen concentration in the atmosphere was 21%. But before that, there was very much low oxygen, or even 0% oxygen in the atmosphere, or I think 1% or 2% oxygen. But there were certain bacteria that were aerobic and they were using oxygen. But the demand of oxygen was greater, but it was not abundant in the environment. So what happens? These aerobic bacteria establish a symbiotic relationship with the eukaryotes. Okay, the cells of the eukaryotes. So what happens? These aerobic bacteria, just to get oxygen, abundant oxygen, they create their symbiotic relationship with the eukaryotic cells. So they were engulfed, they enter the eukaryotic cell and they were using their oxygen. So after that, when these aerobic bacteria enters the eukaryotic cell, so time passes and evolution was there in their structure, and after that it was converted into mitochondria, and it become an essential part of the eukaryotic cell by synthesizing ATP and energy for the cell. So this is known as endosymbiotic theory. In simple, endo means entering, and symbiosis creating symbiosis. So these aerobic bacteria, just to get oxygen, and there was no oxygen in the environment, so they enter the eukaryotic cell and that use the oxygen of the eukaryotic cell. And what they do, they were giving different advantages to the eukaryotic cell. And with the passage of time, there was a change in their structure. And what happens? They were then they become an essential organelle of the eukaryotic cell. Just look here. It is just look the structure of the. If we look to the structure of the mitochondria, so it is just like the structure of the bacteria. Although its size is also the same as bacteria, a bit larger than bacteria, but just look here. It is just like a bacilli bacteria. Okay, so it was consisting its own DNA, because we know that bacteria consists DNA. So, and it was circular DNA. The DNA, the nuclear material that is present in the mitochondria, just look here, this is the mitochondria, it is circular DNA. Okay, and more than one copy of DNA is present inside the mitochondria. So this was a short story about the evolution of mitochondria, that how does it enters the cell and what was the main cause that it enters the cell. Okay, so and this is the microscopic image of the mitochondria. Now here come our main concern is with the mitochondrial DNA. We are not discussing the structure of mitochondria. We are just sticking to the mitochondrial DNA. Look here, this is nuclear DNA and this is the mitochondrial DNA. So look here, it is linear in shape and the mitochondrial DNA is round in shape. Okay. The second part is that this is a small difference between the mitochondrial DNA and the nuclear DNA. The main is that now we will come to the gene or the genotype of the mitochondrial DNA. 2 to 10 copies of DNA per mitochondria. There should be 2 to 10 copies of mitochondrial DNA because I mentioned that there are more than one part of copy of DNA inside a mitochondria, and it is also known as nucleoids. Okay. Now here come that it totally this mitochondria consists of 6,569 DNA bases. Okay, and it consists of 37 genes. These nucleotide bases consist of 37 genes. Just keep this thing in mind. And here the 13 then these genes are also classified into two categories. So the 13 genes code for enzymes that are involved in the respiration process. As I have told you that mitochondria is performing respiration inside our cell and producing ATP. So the 13 genes are coding enzymes that are used in the oxidative phosphorylation. Okay, and the 24 genes code for the transfer RNA. As you know that there are 20 different transfer RNA for different 20 proteins for 20 amino acids. So the 24 genes codes for transfer RNA and the 13 genes codes for the enzymes that are taking place in the process of oxidative phosphorylation. Okay, so just keep these things in your mind. Now secondly, this is a animated structure of the mitochondrial DNA. Just look here. Basically, it consists of two chains. This is the inner chain, and this is the outer chain. It is also known as heavy chain and light chain. So the outer one is known as heavy chain. Okay, this is the heavy chain. It is heavy chain, and the inner chain is known as the light chain. And this is just because of it. It is known as heavy chain because it consists of high amount of guanine bases. Okay, so that's why it is known as heavy chain. And the light chain does not consist high amount of guanine, so it is known as light chain. And as I have told you that it 24 to 22 to 24 genes code for transfer RNA and 13 genes codes for the enzyme that are taking place inside the process of respiration. Secondly, just look here, this portion code for the NADH dehydrogenase subunits. This portion, these are the enzymes. Okay, different enzymes that are taking place in the the respiration. So these are the portion. Okay, this is portion. So you can study it when you pause the video, so you can see it. Okay, after that, okay, one thing in mind is control region or D loop. So I will discuss this D loop in the second part of the video when I will discuss the replication of the mitochondrial DNA. Because if I want to explain it here, so you will not understand it here because this D loop, why it is called D loop. So I will discuss it there. For now, time being, just consider it that these the portion 22 genes are coding for transfer RNA and 13 genes are coding for the enzyme that are taking place inside the respiration. Okay, and this D loop is not coding any gene, any proteins. Okay, so it is not this place is not coding any genes. It is consisting of no genes. Okay, so keep this thing in mind. Now I will discuss the difference between mitochondrial DNA and nuclear DNA. So look here, mitochondrial DNA is just like this round shape, and this is the nuclear DNA that consists of 23 pairs of chromosomes, as you know. So this is a basic difference between the mitochondrial DNA and nuclear DNA. If you look here, So one thing, just to keep this thing in mind, that mitochondria can also be, only mitochondria come from the mother. Okay, the mitochondria, or the mitochondrial DNA are coming from the mother. It is not coming from the father. Although both sperm and egg contain mitochondria, okay, but after the sperm penetrates the egg, its mitochondria are rapidly destroyed. All the mitochondrial DNA is inherited only from the mother. Just look here, this is the father and the mother. So all the infants, the children are getting the mitochondrial DNA from its mother. Why it is taking, I will discuss it in the diagram. Okay, just keep this thing in mind. Now here is the diagram that you will now know that why it is coming from the mother, not from the father. Just look here, this is an egg cell, okay, and this is a sperm cell. And if you know that there in the sperm cell, here is the DNA content, okay, and the mitochondria lies in its collar. The mitochondria lies in its collar. So what happened when it join with the when the sperm interact with the egg? So what happens? It has only to transfer its DNA, nuclear material, into the egg. It has no issue with the other. The total sperm cannot penetrate inside the egg. So this portion contains the nuclear material. So what happens? The nuclear material is transferred inside the egg cell, and this is remain outside the egg cell. Okay, this portion is remain outside the egg cell. So this RNA, this mitochondria cannot enter the egg cell. So then it cannot be transferred to the infants. Okay, so just because of it, it cannot. The mitochondrial DNA, there is a very much small. There is no mitochondrial DNA, and that is coming from the father. But now research are done, and this research indicates that a very minute portion of mitochondrial DNA can come from the father, but it's very much small in amount. So we are considering it as that it is coming from the mother. And this is the main reason that the mitochondria is present in the collar, and the collar cannot penetrate inside the cell, inside the egg cell. So just because of it, it remains outside the egg cell, and the nuclear material is, enters the egg cell, and the process is complete. So just because of it, the mitochondrial DNA can only be inherited from the mother. Okay. Now here come that there is another difference between the nuclear DNA and the mitochondrial DNA. So I want to tell you that our nuclear DNA has the ability to repair itself. It has a repair mechanism, but mitochondrial DNA lacks the repair mechanism. Okay, if there is a mutation inside the mitochondrial DNA, so it cannot correct its mutation. And if there is mutation in the nuclear DNA, so it can be corrected by different repair mechanism, but there is no repair mechanism for mitochondrial DNA. So if there is a mutation in the mitochondrial DNA, so then its mutation rate of mitochondria, that is about 10 times higher than the nuclear DNA, because it cannot repair. So once it is mutated, so it is then transferred to the infants in the mutated form. Okay, so this is also a very basic difference between the nuclear DNA and the mitochondrial DNA. The very much other important difference is the coding difference. The mitochondrial genetic code differ from the code of the nuclear DNA. Here are the codes that are coding different amino acid. But just look here, this just look here, this UAA, UAG, and UGA. It acts as a stop codon in the nuclear DNA. But in mitochondrial DNA, the UGA portion codes for tryptophan. Okay, so there is a difference. Just look here, this AUA, in the nuclear DNA code for the isoleucine, but in the mitochondrial DNA, this AUA code for the methionine. So there is also a slight difference in between these nuclear DNA and the mitochondrial DNA on the basis of these coding. Okay. Now here comes the other difference in the nuclear DNA. As we know that there are exon and introns. Exons are the essential part of the gene, and the introns are the non-essential part of the gene. When they are modified, so after that they are destroyed when the this after the post transcription modification, these introns are removed. Okay, but I want to tell you that mitochondria DNA does not consist of any introns. All the genes will be totally exon. They will code for a specific protein. It will not have any intron. But in the nuclear DNA there are introns, and in the mitochondrial DNA there are no introns. Okay, there are totally exons. Keep this difference in mind. The last difference is that the nuclear DNA is monosystronic, means that at a time one gene can be translated. A messenger RNA can translate. When it is translated, it should produce a single protein. But in case of mitochondrial DNA, it is polysystronic, because a single messenger RNA can code for several proteins. So there is a this is also a special difference in between it. The nuclear DNA is monosystronic, means when the messenger RNA that is produced after the transcription process, it could code only a single protein. So it is known as monosystronic. Mono means one. Okay, but in mitochondrial DNA, when messenger RNA is produced, so this messenger RNA at a time can code for several proteins. So these are known as polysystronic, and this is also the same in the bacteria. Okay, in the prokaryotes. Okay, so as now you will know that first of all this mitochondria was a eukaryote, because different feature of the prokaryotes are the same as the mitochondria. So these were the difference between the nuclear DNA and the mitochondrial DNA. We discussed the differences. We discussed the main reason that why mitochondria can only be inherited from the mother, not from the father. The main cause we have discussed it. We have discussed the structure of the mitochondrial DNA and the genes that are the proteins and the transfer RNA they are coding, the portions that which the different portions that encode for different enzymes. We discussed the endosymbiosis theory, that what basically is an endosymbiosis theory, and how this mitochondria come into being. Okay, so this was all about the today's lecture. It was an introduction. In the second part, I will discuss the replication of the mitochondrial DNA, because it is very much unique in nature. So the replication is also a type a bit unique in nature. Okay, so if you have understand this lecture, so like this video, share it with your friends, and subscribe to my channel, so in order to get the notification of other interesting and conceptual videos. And thank you very much. God bless you.